Btric News. I'm Juan Felipe Castillo, the presenter. We have Santiago Campos as an expert on the functions of the dogma in technology, and we have Juan Castro as the interviewer. But what is a central dogma? The central dogma is a process in which the DNA information is converted into a functional product. It was first used in 1958 by Francis Crick, the discoverer of the DNA structure. So, the central dogma explains that the flow of genetic information from the DNA to the RNA to make a functional product, a protein. Also, the central dogma of biology suggests that the DNA information is necessary to pass to the RNA messenger that later is going to pass to the ribosomes. Also, the ribosomes are like the factories of the proteins because they use the DNA information that later pass to the RNA information. They convert the code into the protein. What is the gene expression? So, the gene expression is the process in which the DNA is converted into a functional product as a protein. So, the gene expression is divided in two parts. First one, the transcription, and second one, the translation. So, for the transcription is when the DNA in a gene is copied to produce an RNA transcription called RNA messenger, or mRNA is the same. So, this information is carried out by an enzyme called RNA polymerase, which this uses the available nitrogenous bases from the nucleus of the cell from the mRNA. So, with this information, they change the thymine to an uracil. So, now is the translation. The translation occurs after the messenger RNA has carried the transcript message or code from DNA to the protein factory. In this case, obviously, is the ribosome. So, the message carried by the mRNA is read by a carrier molecule called transfer RNA, like the tRNA is the same thing. So, the mRNA is read 3 letters, that is a codon. So, each codon has a specific amino acid. For example, the 3 bases GGU code from the amino acid called glycine. So, actually, the count of amino acids began with the med, that is the AUG, and obviously, finally, with an stop. So now, Santiago Campos is going to explain the functions or the uses that have the dogma now in the present.
Hello, I am the reporter Santiago Campos, and today I will come you to talk about the molecular dogma in the biology that we can apply in the health of the humans. The central dogma of molecular biology refers to the 3 processes carried out by the nucleic acids, both DNA and RNA: replication, transcription, and translation. It illustrates the mechanism that transmission and expression of genetic in a brain. The molecular theory in the biology of health can be seen as one of the greatest achievements of molecular biology in the 20th century, was the power, in a directed manner, to manipulate the genetic information of organisms, including men. The technological approach, called the dominant DNA technology, has allowed a fundamental advance in the way in which the functioning of genes currently understood, the possibility to correcting genetic defects and the production of new therapeutic alternatives, all in order to improve the global health. However, despite the great advances in these areas, there are the potential risks that led us to question how these advances should be used and regulated to have biosecurity protocol, in addition to being able to give the inhabitants of the planet equal opportunities for use. Molecular biology has different fields of applications. It is applied of almost all productive sectors, both for use for human consumptions. In the food sector, it stands out the production of dyes, gums, organic agents, flavor enhancements, flavorants, acidulants such as citric acid, enzymes, additives, and etc. In the sweeter sector, for example, the protein tomatin, extracted from a fruit, has already been expressed in several microorganisms. In the agricultural sector, the domestication by men of plants for agricultural production was a long process that had evolutionary consequences in many species. In the livestock sector, it has contributed to more rapid growth in the size of some fish species, to a higher production of milk from cows, or the quality of the milk of animals such as pigs. This can be achieved through the use of hormones in the growth phase of livestock. In the pharmaceutical sector, molecular biology allows that more effective drugs that can develop and they cause the less adverse effects. Molecular biology is also applied in forensic medicine. The determination of genetic fingerprints is another application to molecular techniques, specifically PCR and restriction enzymes. These techniques are currently applied in forensic medicine and the police investigation in order to identify individuals from biological samples such as blood, semen, saliva, skin, and hair, found in cases of death, rabies, and other type of aggression. The profiling of DNA allows the identifications such as evidence with a degree of certainty was previously considered impossible, since it's possible to compare DNA samples to check whether they belong to an individual or not, or there is a kinship between them. Similarly, the technique can provide evidential evidence in case of claimed or unrecognized paternity.
Now, Juan Castro is going to interview an expert on the topic.
Hello, my name is Juan Sebastián Castro. I am the interviewer from the news Btric. I go to interview the student of school, San Viator School. His name, Gabriel Felipe. He is in science and health, and he tell me about the biotechnology. I made 5 questions from the information that give my teammates in the past videos. Okay, let's go. Hello, I am with Gabriel Felipe.
Hi.
Okay, what do you know about biotechnology?
Well, I know that it's a very big camp. It covers from the nanotechnology, from the non-used prostheses to recuperate, for the recovery or people that have lost an arm or a leg or an extremity.
Okay, can I make from you for some questions?
Yeah, for sure.
Okay, first, what do you think about the use of biotechnology in the animals?
Well, I think that it might be polemic, but in some cases, with this technology being less invasive, it could be, yeah, a good method for research, but not like some labs that were discovered that they were almost torturing the animals.
Okay. Are you use or see the use of biotechnology?
Yes, I have seen in lot of people that have prostheses and a lot of documentals and newspapers talking about the prostheses and nanobots, nanotechnology.
Okay. Do you like that the humans use biotechnology to change the DNA?
Yeah, it could be good, for example, in some countries, they're checking DNA. For example, one case is Jennifer, no, Angelina Jolie, that checked her DNA, and from that she knew that she would have mama cancer at some age, but how she detected it, even after it happened, she could remove it and she didn't have any problem in those years. But change the DNA could be polemic with the religious people. Why? Because it plays with the myth of trying to be God, trying to play to be God. So it could be a polemic topic that I, from the science camp, can answer, but in some cases, yeah, I agree with the techniques.
Okay. Do you think that is good that use biotechnology in the food that we eat?
Yes, yes. I am so sure that with a good revision and a good administration, it could even save the planet. For example, corn. Do you know the corn? Yes. They made a corn that is invulnerable to insects, to some diseases that could kill that plant, and to grow faster. So with this type of DNA change foods, they can feed a lot of people. But also in Colombia, it's seen a model that sells the apples, the red apples, the pretty apples that you see at the stores. They boil the seeds, they change a little bit the seed for the apple to look full red, and they say to the farmers, this is the only seed that you could sell, but it is more expensive for you, and you will sell it less expensive than the other apples. So a lot of farmers are being threatened with this gene.
So now see you later in another transmission.